Hi, this is Neuron Loom, and now we're going to discuss how acetylcholine actually causes vasodilation. We all know that acetylcholine is a major neurotransmitter in the human body which causes transmission of neuronal signals. It has various actions. It mediates action through two receptors, muscarinic receptors and nicotinic receptors. Vasodilation is mediated through muscarinic receptors. So let's take a look at how actually vasodilation occurs. So this is endothelial cell of blood vessel. On the membrane of this endothelial cell, there is muscarinic M3 receptor. On stimulation of this M3 receptor by acetylcholine, there is stimulation of another enzyme which is phospholipase C, which converts phosphoinositol diphosphate into inositol triphosphate and diacylglycerol. Now this inositol triphosphate stimulate endoplasmic reticulum to release calcium ions. These calcium ions will form a complex with calmodulin, that is calcium calmodulin complex, which further stimulates endothelial nitric oxide synthase enzyme, which converts arginine, that is an essential amino acid, into nitric oxide. Now this nitric oxide synthesized inside the endothelial cell diffuses into adjacent smooth muscle cell and activates soluble guanyl cyclase, which further converts GTP into CGMP. Now this CGMP will activate another enzyme, that is protein kinase of CGMP. This protein kinase G will continue its action through two mechanisms. First of all, it will activate myosin light chain phosphatase. Let's understand that phosphorylated myosin is only active form of myosin. We all know that myosin is an essential protein for the contraction of the muscle. And when phosphatase enzyme is getting activated, there is definitely dephosphorylation, which reduces the availability of phosphorylated myosin, and therefore there is decreased contraction, which eventually means that there is relaxation. Apart from that, this protein kinase G also inhibits calcium membrane channels. So what happens when there is no inhibition? That calcium from the periphery through this membrane channels enters inside the cell and forms calcium calmodulin complex, which results into the contraction of the cell. But now, because there is inhibition of this whole system, there is relaxation. Okay, now what I want you to observe is the duality of this calcium calmodulin complex, right? See, in the endothelial cell, this calcium calmodulin complex is causing the release of vasodilator by stimulating nitric oxide synthase enzyme. Here it is promoting vasodilation, but in the smooth muscle cell, what is it doing? That it is promoting contraction of the muscle. That's the duality of this calcium calmodulin complex, and it is the most astonishing part of this whole mechanism, and that's my favorite part. Okay, apart from that, we all know that in some conditions like hypertension, atherosclerosis and diabetes, there is vessel damage, and when there is vessel damage, there is damage of this endothelial cell, and because of that, this acetylcholine directly diffuses inside the smooth muscle cell. Containing that, this smooth muscle cell also have muscarinic receptors on its membrane. So on direct exposure or direct diffusion of acetylcholine, this muscarinic receptor gets stimulated, and it further stimulates phospholipase C enzyme, leading to synthesis of IP3, which will further stimulate or induce sarcoplasmic reticulum, which really increases the intracellular calcium concentration, and this will lead to contraction of the smooth muscles. So in healthy adults, in healthy arteries, there is vasodilation on exposure of acetylcholine, but in patients with vessel damage, there will be contraction of this smooth muscles. Okay, So yeah, that's the concept of the today, and thank you.